Hello everyone, my name is Sriram Yadav from Department of Biotechnology, IIT Roorkee. I welcome you all for this course of Plant Developmental Biology. For this course, I was assisted by Mr. Tushar Garg, a PhD scholar in my laboratory. Coming to the introduction of plant developmental biology. So, in plants or any higher organism, growth and developments are very important and essential process. And plant developmental biology is a study of how various tissues and organs are developed, starting from a single cell zygote in a complex multicellular plants, how different organs are developed during various phases of their life cycle. So, this course will provide an overview of mechanisms underlying the primary meristem establishment and maintenance, cell specification and differentiation during various organ development in flowering plants. In general, if you look the life cycle of a land plant, it has two major phases, sporophytic phases and gametophytic phases. So, sporophytic phases basically it is diploid phase of life cycle, and it is started from a single cell which is called zygote. This zygote, which is generated by the fusion or fertilization of male and female gametes, and after this fertilization this diploid zygote cells undergo the process of cell division, typically mitosis cell division, to generate multicellular sporophytic phase. And then at some point of time in their life cycle, this sporophytic phase undergo the process of meiosis, and this meiosis generates many haploid spores, and these haploid spores, when they undergo the process of further cell division, mitotic cell division, and they generate multicellular gametophytic stage, and this entire process is called alteration of generation in land plants. So, if you look the overall evolution of land plants, so the land plants, they belongs to streptophytes clade of plant kingdom, and this streptophyte clades, it includes caryophytes, which is green algae, and embryophytes, which is typically called land plant. And one important thing is that in caryophytes the sporophytic stage is unicellular, whereas in all other land plants this is multicellular. In embryophytes, this can be further divided into vascular plants and non-vascular plants. So, the bryophytes, which belongs to the non-vascular plants, and then lipophytes onwards, they belong to the vascular plants. And then at some time point during the evolution, particularly from here, there is a true leaf formation, and then later on there is evolution of seeds like structure. So, if you look the event of development of the plants, so very important events, one is the multicellularity in the sporophytic, and then the vascular tissue. These vascular tissues are very important because they forms the major transport system in land plants or in higher plants, and it is very important for land plants since they are terrestrial, they become sessile. It is very important for them to have a very robust transport system required to take minerals and waters from the soil. We will be mostly focusing our developments on the angiospermic plant. Further, if you look the alteration of generation across the land plants, so there are few important things which you can see. So, in bryophyte, the major part of the bryophyte life cycle is the gametophytes, and sporophyte, which is dependent on the gametophyte. But in fern onwards, the major component, the large form, is the sporophyte, and gametophyte is basically a small independent form. But in seed plants, which includes basically gymnosperm and angiosperm, it has a reduced gametophyte, which is basically dependent on the major large sporophytes. So, during this evolution, there is, if you look the duration of their life cycle between sporophytic versus gametophytic stages. So, mosses, which is having more gametophytic or more duration of the gametophytic, less duration of the sporophytic, whereas if you go across ferns, gymnosperm, angiosperm, the gametophytic stages are getting restricted and the sporophytic stages are getting expanded. Now we will discuss about the developmental events during angiosperm's life cycle. Typically, an angiospermic plant has two major parts. One is the root system, another is the shoot system. And as we discussed earlier, a sporophyte is a dominant phase of the angiosperm life, and this entire sporophyte can be divided into three major phases. The one is the embryo development, then vegetative development and reproductive development. The embryo development starts after the process of fertilization, and then it terminates by the seed dormancy. Then vegetative phase starts when seeds are germinating, and then during the vegetative phase all the vegetative organs are formed, and then at a certain time point there is a transition, there is a phase transition which is called floral transition, where vegetative phase get converted into the reproductive phase, and during reproductive phase the reproductive organs like flowers are developed, and in this flower there is a sex organs which is basically anthers and carpel, and there the process of gametophyte is restricted in case of angiospermic plant. So, if you look the embryogenesis and seed development process. So, actually during this embryogenesis process the basic body pattern is established before the seed dormancy. So, in overall throughout the embryogenesis process, four major things are happening. Number one, the apical basal axis polarity is established. Number two, radial tissue patterning occurs, which occurs through the spatial organization of the tissues, particularly epidermis, ground tissue and vascular tissues. Third important things what happens is the positioning of shoot and root apical meristem along the apical basal axis, and finally there is a formation of cotyledons. So, if you look embryogenesis process, so embryogenesis typically begins with an asymmetric cell division of zygote which produces a small terminal cell and a large basal cells. The terminal cells makes proper embryo and basal cells produces suspensor. Eventually the suspensor generates a stalk or a filament which attach proper embryo to the maternal tissues. At this stage, if you look the shape of embryo, it looks like globular, and this stage is called globular embryo. During globular embryo stage, the three major body tissues are established. The outermost layer which is called epidermis or protoderm, then middle layer is ground meristem or ground tissues, and the innermost layer is procambium. Then next things what happens, cotyledon primordia initiates, and this results in the change of the shape of the embryo. Now it becomes heart shape, and further this heart shape get converted into torpedo stage embryo, and this is the stage around which the shoot apical meristem and root apical meristems are actually positioned at apex. Shoot apical meristems are positioned at the apical site and root apical meristems are positioned at the basal site. This is a mature seeds where you have a seed coat and proper embryo which is attached with the suspensor, and in this embryo we have the basic body plan, for example root apical meristem, shoot apical meristem, provascular tissues, and at this stage this embryo undergo the process of dormancy and it can stay for long period of time. The embryo breaks their dormancy during seed germination, and which requires a proper external cues such as temperature, moisture and lights. Seed germination require extensive cell elongation. Depending on the region of elongation, the pattern of germination can be categorized into two types. One is the apical germination and hypogeal germination. In apical germination, elongation occurs in the region between cotyledon and the radicle, which is also called hypocotyle, and cotyledons comes above the ground. In hypogeal type of germination, when elongation occurs in the epicotyl region, which means that region between the cotyledons and shoot apical meristem, the cotyledons remains below the ground in this case. This is the typical example of apigeal germination and hypogeal germination. As you can see here, the region, the hypocotyle region, when it elongates, the cotyledons are coming outside. In case of hypophysial, when the region between shoot apical meristem and cotyledons is elongates, so there is the cotyledons remains underground. Next developmental stage is vegetative developmental stage. This starts with the seed germination, and once you put seeds for the germination, plants undergo mostly post embryonic vegetative development. During primary vegetative phase, following developments occurs. First very important thing is the primary growth, which occurs at the apical meristem, both shoot apical meristem and root apical meristem. For having continuous growth, it is very important that plant always maintains both the meristems throughout their life. Next, during vegetative development is the vegetative organ formation. The process is called organogenesis. Slightly later stage or at the same stage, you will also have a process of organ patterning, where special tissues are patterned in a proper configuration, and vascular tissues development, which is also a part of tissue patterning, occurs simultaneously. So, if you look at typical example of root development, so this is a typically growing primary root. The primary root can be divided into 3 zones. This is the meristematic zones, this is elongation zones, and this is the maturation or differentiation zone. And root apical meristem is positioned at very tip of the growing root. And the important process which is root apical meristem maintenance, and this basically generates, this has the stem cells which generate the cell lineage for tissue patterning later on. In higher region, tissue patterning occurs, where you can have a clear different layers of the tissues arranged in a proper pattern. In differentiation zone, one of the very important process occurs, which is called root branching. In root branching, lateral roots are formed. You can see lateral root primordia are initiated, first specified, then initiated, and then finally lateral root primordia emerged out. Similarly, if you look the shoot development, during shoot development, the stem cell maintenance occur in the region which is the shoot apical meristem. This is the region where stem cells are maintained, and when this stem cell, when the cells leaves the meristem in the peripheral zone, it undergo the process of differentiation. During vegetative phase, the major organ which forms is the leaf. You can see P1 is the first leaf primordia, second leaf primordia, third leaf primordia. So, during shoot development, the important process which takes place is the root shoot apical meristem maintenance, organogenesis, tissue and organ patterning, and then shoot branching. Shoot branching normally occurs through the axillary buds, and of course, vasculature is equally important. So, once this vegetative process is completed, vegetative development is completed, a major transition occurs in the life cycle of an angiosperm plant, and then transition is from vegetative phase to reproductive phase. Lot of internal and external factor regulates this process, but once the decision is taken place, the vegetative phase transit and it start making the reproductive organs, which are basically flowers, and flowers are typically arranged in form of inflorescence. So, if you look at the meristem level, so during vegetative phase, the structure which is responsible for the growth is shoot apical meristem, but at the time of transition, the identity of this shoot apical meristem is changed. Now it becomes inflorescence meristem. The shoot apical meristem, it was making leaf at the flanks, but once its identity is converted to the inflorescence meristem, the flanking primordia is now floral primordia. So, if you look at typically growing inflorescence, you will have lot of flowers, and then the flowers will have floral organs, which is sepal, petals, stamen and carpels. And if you look, the stamens and carpels are very special tissues, are very special cells of stamen and carpels. They have a restricted gametophytic phase. If you look this plant, so this is a typical flower, this is typical angiospermic flower. In flower there are 4 organs mainly, the sepals, petals, stamen and carpels. The stamen is male reproductive organ, carpel is female reproductive organ, and then The process of gametophytic stages initiate from here when anthers, which is a male part of stamen, produces pollen grains, and carpels, which has ovule. When they undergo the process of fertilization, they makes a seed which contains embryo, and the seeds has all the information, all the basic body plan in embryo, and when we take the seeds, germinate it, it makes the complete plant. So, the process of gametophytic therefore can be divided into two classes. One is the megasporogenesis and microsporogenesis. This is flower, which is a reproductive organ. So, as I said earlier, flower has sepals, petals, stamens and carpels. Stamens and carpels are the reproductive organ, whereas sepals and petals, they are accessory organ. So, though sepals and petals, they do not directly involved in the process of reproduction, but they play very important role in successful completion of the reproduction. And in a flower, all these organs are properly organized and arranged. So, they arrange in form of whorl. So, during the development, it is not only the organ, it is not only the identity, but their patterning is also equally important. So, if you look, sepals, they makes the first organ, then petal, stamens and carpel, and we will also see in this course that how this floral organ patterning is happening. Now, during the gametophytic phase, so we will look how female gametophyte developments occurs. So, if you look the female reproductive organ, which is carpel in Angiosperm, the carpel has three regions: the stigma, style and ovary. In ovary, there is a structure called ovule, and in ovule we have a megaspore mother cell. So, this megaspore mother cell, which is diploid in nature, it first undergo the process of meiosis to generate 4 megaspores, 4 haploid megaspores, and then during later stages 3 of these megaspores they degenerate, only one remains as a functional, which is called functional megaspores, and these functional megaspores undergo the process of 3 round of mitosis without cytokinesis to generate a cells with 8 haploid nuclei, and then later on these haploid nuclei they get rearranged and they make this structure called embryo sac. Embryo sac development, so this entire process of embryo sac development can be clearly divided into two groups or 2 stages. The first stage is megasporogenesis, where megaspore mother cells divides through the meiosis to generate a haploid functional megaspores. Then another stage is megagametogenesis, where these megaspores undergo the process of mitosis, cell division and rearrangement to generate a female gametophyte. And if you look female gametophyte, this female gametophyte, which is also called embryo sac, it contains 3 antipodal cells on the apical region of embryo sac. Then it has 2 synergist and 1 egg cells at the basal end of the embryo sac, and then a central cell containing 2 haploid polar nuclei. Similarly, if you look the male gametophyte development, so as I said that the male organs are stamens. Stamen has filaments and anthers. This is a cross section of anther. Here we have microsporocytes, which is diploid in the chromosome content, and these microsporocytes, when it undergo the process of meiosis 1, it generates 2 haploid nuclei, then meiosis 2, and it makes a tetrads. These tetrads release, and then later on these microspores they undergo the process of another round of mitosis, and they generate 2 asymmetric cell. One is called vegetative cell, another is called generative cells. Later on these generative cells migrate in sites and then makes a kind of cell within cell appearance. Here there are 2 categories. In first category, the generative nuclei, they undergo the process of mitosis 2 and generate 2 nuclei before the germination or before the pollination. But in second case, they remains in this form, and when they get attached with the stigma of carpel, then this division takes. But in overall, because of these divisions, there are 2 sperm cells are generated. Next stage of the life cycle is pollination and fertilization. So, once the pollens are ready, so this is a typical germinating mature pollens which has 2 sperm cells and 1 vegetative cell nucleus, and this is mature ovule cells. So, in ovule cells you can clearly see there are antipodal cells, there are synergist, egg cells and polar nuclei. But the pollination is a process through which the pollen grains they get attached with the stigma of carpel and then they germinate, but it is not always the case. So, all the pollens cannot fertilize or cannot pollinate with any kind of carpel. So, there is some self incompatibility issue. So, if pollens are not compatible with the carpel, they may not result in the successful pollination, where either their germination itself is inhibited, or even though if we start germination, but they cannot reach to the ovule and they cannot fertilize the embryo sac. But eventually what happens in the case where the fertilization is successful, the pollen germinates and it makes a structure called pollen tube. When pollen tube reach the micropyle of the ovule, there is, if you look here, it is more clear here, the one of the synergist cell it secreted some kind of signals, and this signal is very important for pollen tube to reach to the embryo sac. And once it reaches to the embryo sac, it releases 2 of the sperm cells. One sperm cells it goes and fertilizes the egg cells and makes a diploid zygote. Another sperm cell it goes and fuses with the polar nuclei, 2 polar nuclei, and it makes a triploid cells, which eventually generates the endosperm. And that is why this process is called double fertilization, because there are 2 rounds of fertilization occurs. So, at the end, if I summarize this life cycle of angiosperm plant, so as I said that flowers are the organs where we have the reproductive organs, male reproductive organs as a stamen, female reproductive as organ as carpel. The male reproductive organs they generate microspores or pollens, haploid pollens through the process of microsporogenesis. Female reproductive organs they generate embryo sac which contains egg cell and polar nucleate through the process of megasporogenesis, and then the process of fertilization or process of pollination completes the double fertilization, and then these entire structures are developed as a seeds. So, life cycle of angiosperm plants. So, it is evident that angiosperm plants alternates their life cycle between diploid sporophyte and haploid gametophyte. Sporophyte is an extensive phase, whereas gametophyte is more restricted. Male gametophyte develops in anther of the stamens and contain haploid sperms. Female gametophyte is produced within ovules of the carpel and contains haploid egg cell, 2 haploid polar nuclei, 3 antipodal and 2 synergist cell. Double fertilization is characteristic feature of an angiosperm, which occurs when pollen tube discharges 2 sperm nuclei into the female gametophyte. One sperm fertilizes the egg cell to develop a diploid zygote, another combines with 2 polar nuclei to produce a triploid endosperm. So, this is all for this class. In next class, we will discuss about characteristic of plant development. Thank you.